The idea that diet can influence gene expression, effectively switching cancer-related genes on or off, has captured public imagination and scientific attention alike. This concept is rooted in the field of epigenetics, which studies how environmental factors, including food, affect the way our genes function without altering the underlying DNA sequence. Unlike genetic mutations, which are permanent, epigenetic changes are potentially reversible. This opens a compelling door. If certain nutrients or dietary patterns can shape gene behavior, could food truly play a role in preventing or modifying cancer development? While the science offers intriguing possibilities, the practical application is still in its early stages, and overstating its effects risks misleading patients about the complexity of cancer biology. Epigenetic modifications occur through several biochemical processes, such as DNA methylation, histone modification, and the regulation of non-coding RNAs. These mechanisms determine how tightly DNA is packaged and how accessible certain genes are to be turned on or off. In cancer, normal epigenetic regulation often goes awry. Tumor suppressor genes may be silenced, while oncogenes become abnormally active. The question is whether specific dietary compounds can intervene in this process, correcting or preventing such dysregulation. Some nutrients and phytochemicals have shown potential in modulating these epigenetic processes. Folate, a B vitamin found in leafy greens, legumes, and fortified grains, is a critical donor in the methylation process and plays a role in DNA synthesis and repair. Inadequate folate can lead to DNA instability, a known cancer risk factor. However, excessive folate, especially from synthetic supplements, has also been associated with increased tumor progression in certain contexts, particularly in people with existing precancerous lesions. This duality illustrates the risk of viewing any single nutrient as universally protective. Sulforaphane, a compound found in cruciferous vegetables like broccoli and Brussels sprouts, has been studied for its ability to influence histone deacetylase, HDAC, activity, which affects how genes are packaged and expressed. Preclinical studies suggest it may help restore the function of silenced tumor suppressor genes. Similarly, polyphenol like curcumin from turmeric or epigallocatechin gallate, EGSG, from green tea have demonstrated anti-inflammatory and epigenetic modifying properties in laboratory settings. Yet most of these effects have been observed in isolated cells or animal models, where dosages are often far higher than what humans can consume through diet alone. That's the key limitation. Promising results in cell culture don't automatically translate to real-world outcomes. The human body is far more complex, with thousands of interacting compounds and metabolic pathways. It's also unclear whether dietary epigenetic modifications are sufficient in strength or duration to meaningfully impact cancer initiation or progression. And even if some nutrients show benefits, they do not act in isolation. The effect of any single food compound must be considered within the context of the entire diet, lifestyle, and genetic makeup of the individual. There is also a growing concern about the commercialization of epigenetic diets, often promoted with exaggerated claims and unsupported protocols. Some companies now offer DNA testing services that recommend specific foods or supplements based on alleged epigenetic profiles. These services rarely have robust scientific backing, yet they play into the seductive idea that we can hack our biology through food. For cancer patients or those at high risk, this messaging can be misleading and may divert attention from more effective, evidence-based interventions. Diet certainly has a powerful role in shaping long-term health and may influence cancer risk, but it should not be mistaken for a precision tool capable of selectively reprogramming genes on demand. While ongoing research into nutrigenomics and epigenetics is promising, we are far from the point where specific foods can be prescribed to turn genes on or off with measurable results in humans. Overemphasizing this potential may give patients false hope or push them toward restrictive or costly dietary regimens with little proven benefit. Ultimately, the most meaningful impact of diet on gene expression is likely to come from consistent, long-term patterns rather than isolated compounds. A diverse, plant-rich diet that includes a variety of fruits, vegetables, whole grains, and healthy fats may support overall genomic stability, reduce chronic inflammation, and promote metabolic balance, all of which contribute to cancer resilience. But the idea that a glass of green tea or a serving of kale can epigenetically silence cancer genes is, at this stage, more hopeful narrative than proven fact. Diet matters. But in the context of cancer prevention and treatment, it matters in conjunction with not instead of genetics, environment, medical care, and lifestyle. The promise of epigenetics lies not in magical switches, but in understanding the many small, cumulative ways our habits may help shape long-term cellular behavior. That power is real, but it should be respected, not romanticized.